right, so if I have an individual who has, let me do this here, who has blood type A negative, all right, what antibodies would they have in their blood? B. All right, with the possibility of having D if they had been exposed via transfusion or giving birth to a kid that was positive, okay? So when you're asking yourself what type of blood can this person receive, that's the first question you want to ask, is what antibodies do they have? Because remember, you never give antigen, you never give antigen to the matching antibody. So if the person has B antibodies, then we cannot give any blood type that has a B antigen. Okay. So before I go any further, do you guys understand antigens and antibodies? All right, we'll talk about antibodies again next week. Antigen is the marker on the cell surface, okay? An antibody is the protein that matches that marker. So if I have blood type A positive, I would have antibody B. Would I ever have antibody D? No. No. All right. So here is, let's just make sure, here we've got a cell that is A negative, would have on it A antigens. Okay? A blood cell that is A positive would have the A antigen. And the D antigen. Okay? Now, both of them would have in their blood antibodies against B. All right? So they're going to have antibodies that would recognize the B antigen. Now, what would happen if I gave the person with A negative blood A positive blood? The actual steps in the process we'll cover on Thursday next week, when we talk about B lymphocytes. But the A antigen will be fine, right? There is no B antigen for the B antibodies to bind to. But if we have this blood type given to this person, a B lymphocyte that had B cell receptors that matched that shape of the D antigen would be triggered to become a plasma cell, and it would start synthesizing antibodies against the D antigen. Okay? And now this person would have antibodies in the D antigen against the D antigen, so that if it was given another transfusion that had the D antigen, these D antibodies that were now present in the A negative blood would agglutinate, would recognize and bind to the D antigens of that incoming blood. Okay? So if they got another transfusion of A positive blood those cells would be agglutinated. All right, now, so I can receive, if I am A negative, I can receive any blood type that doesn't have the B antigen. So I can receive my own blood type, obviously. I can't receive B. How about AB? No. All right, O?
Yes.
As long as it was negative. Now O is going to have antibodies coming into my blood, and those antibodies can recognize my A antigen. So it's a matter of dilution. Okay, the incoming antibodies are not as devastating as the antibodies I already have because there's fewer of them, and they're diluted, and while they might agglutinate a few hundred, they're not going to agglutinate a million. Okay? And so it's okay. It's not what we do if you know you're going to have surgery. So if you know you're going to have surgery, you normally go in a few months early, a couple, 6 or 8 weeks early, and they'll take blood and they'll store it for you, give you time to make your own back again. Or if it's something that's not that planned that early, they will draw blood from you, type it similar to the way we did it in lab, type from the blood bank the blood that has the same type, and then they will mix the two before they give it to you to make sure there aren't any other factors that are incompatible that were missed, or that the wrong typing was done accidentally. Okay? So you wouldn't normally be given O negative as your transfusion after surgery. You would be given your blood type A negative. All right? But if you are coming into the emergency room and you've been in a traumatic accident and your leg is cut off and your blood pressure is dropping and all they have is O negative, they can give that to you. Right? They won't give you A positive because that's just going to make matters worse. And what they give you is of no use because it's going to become agglutinated. Okay? Now, what can you donate to? Again, the thinking is you don't donate to anybody that has antibodies against you. It's always the receiving blood that we're looking for the antibodies and making sure that those aren't going to be affected. So my blood type is A. So I can't give to anybody that has A antibodies. All right? So I can give to A negative. Can I give to A positive?
Yes.
Yes. Because A negative is nothing. All right? There's nothing D-wise that has to do with that. So I can give to A positive. AB. Can I give to AB? Okay, does AB have any antibodies against A? No, it doesn't have any antibodies at all. Okay? So I can give to AB, positive or negative. Can I give to O? No, because it has both A and B antibodies. Okay? So you see how you can look at that? So you're always looking at the antibodies that are present in the receiving pool, whether you're receiving or donating. You're looking at the antibodies that are present in the receiving pool, okay, or the receiving individual. All right, now let's look at, any questions on this specifically? Okay. Now let's look at questions, word problems. So Steve has A and B antibodies in his blood. To whom, use the proper English here, so who could he give blood to?
Oh.
So first of all, we don't have to figure out his blood type because we know what antibodies, well actually we do. But we need to know what his antigen is so we know who he can't give blood to. So if he has A and B antibodies, what's his blood type? O. Question mark. Okay? Because remember, if we don't have D antibodies, then we don't know whether he's positive or negative. Right? All right. So, since he has A and B antibodies, but he doesn't have any antigens, he can be given to O blood. Because O's not going to have antibodies against him. I mean, it has antibodies, but he doesn't have the antigen for them to accumulate to. He can give to A, to B, and to AB. Positive or negative? Only positive, okay? Because we don't know if he's positive or negative. And since we don't know, we don't want to be giving positive to negative. Positive can receive either, so just to be safe, he should only give his blood to positive. That's all we know about it.
But if he has antibodies against A and B, how can he? Oh, okay.
He has the antibodies. Okay, so if we change it to what blood type can he receive, now he has A and B antibodies. So he can't receive any blood type that has A or B, right? So that means he can only get O. And this time, he should only get O negative. Because he might be O negative, and we don't want to give him O positive. Okay? So to be on the safe side, he should only receive O negative, because remember, that's our universal donor. Okay? Since we don't know whether he's positive or negative, we don't want to jeopardize that. Alrighty?
But if we know that he has O blood, we're not sure what it is, then how come we're giving positive blood to?
So he can donate only to positive. Because if he's no negative, that's a universal donor. Donating O negative to positive isn't going to do any damage. And if he's O positive, there's no problem either. We don't want to give him to negative, because if he's O positive, that could trigger an immune response,
okay.
and they may have antibody.
So you can give negative to positive, but you can't give positive to
Correct.
negative.
Okay. All right, so you might see questions like that where you have to kind of work backwards from things. So let's do an RH question, RH negative erythroblastosis question. Okay. So the mom is A positive. Her baby is A negative. Any problem? Mom is A positive. The baby is A negative. If the baby's blood gets into the mother's blood, it's not going to matter. Because the baby has A antigen, the mom has A antigen. There's no incompatibility there. The baby doesn't have the D antigen, so if his blood gets into the mother's blood, there's not a problem. Since the mother has the D antigen, she's not going to have a D antibody. So when her antibodies cross into the baby's blood, only D antibodies will cross into the baby's blood, and the baby doesn't have the antigen.
What about the D antigen in the mom's blood?
What about it?
What if the baby gets that?
Mother's blood, the antigen itself, the blood cells won't cross into the baby's blood. It's the antibodies that can cross the amniotic membrane or the capillaries. Okay? When this crossing normally occurs when the baby's blood gets into the mother's blood is after it could ostensibly, it's after the placenta is separating, okay, and the baby's gone. Okay? So everybody kind of get where that's coming from? So the only time we have a problem that might even occur is if the mom is, of course, that could be A, B, or O. It doesn't really matter. So mom is A negative, okay? And let's say the baby is A positive. The baby's A negative, is there a problem?
No.
No. Okay. But if the baby is A positive, two things. If it's really, 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 really her first pregnancy, not just her first baby, the baby will probably be okay if she's never had a transfusion, okay? Because the mother's sensitivity will develop after this baby is born, and blood cells from this baby have been left behind in the mother's blood. If this is her second or third or fourth baby who's positive, then antibodies from the mother will start clumping and agglutinating the baby's blood, and that's what we have, erythroblastosis fetalis. Okay. So let's say that this is her first baby. She didn't have any prenatal care. She's at the hospital, the baby's born, they do her blood type in case she needs a transfusion, it's A negative. So they're concerned about what the baby might be. Baby's fine, but they do the baby's blood and it's A positive. So now they've decided that she's going to get RhoGAM. So what is RhoGAM?
Stops the antibodies from.
Okay, so it's antibodies against what? Anti-D. But the mother's blood type doesn't have any D antigens. So what is the anti-D doing in the mother's blood?
Seeks out and destroys the baby's blood.
It's looking for fetal red blood cells and agglutinating them. Are they going to harm the mother?
No.
No. So we're not trying to prevent a transfusion reaction in the mother. What we're trying to prevent is what's called the primary immune response. That first exposure, what I drew over here earlier, where the lymphocytes become plasma cells and are primed to start producing the antibodies. So we want to get those fetal red blood cells out of her bloodstream before she completes that process and now has memory cells that every time she has another child that is positive will spike that activity and start sending antibodies across the placenta to the baby or across the amnion to the baby. Okay? All right, one last exercise. Almost wrote NDC. All right, if I'm going to do this a little bit backwards. If our blood type is A positive, what will my typing tray look like? Rather than putting it up there and having you tell me what my blood type is. So my blood type is A positive. Where will I see agglutination occurring?
A.
Okay, A and D and nothing here. Okay? Yes?
On the exam, will you ever do it like the other way, where it would be like, instead of the antigen, you'll have the antibody? You know what I'm talking about?
Only in that word question,
In the
where I
blood.
tell you we don't look for the antibody in the blood.
Oh, okay.
We're always looking for the antigen
Okay.
when we're doing typing.
Okay.
The only time I would ask you the blood type based on the antibody is the word question, where I say so-and-so has antibodies, what's their blood type? That thing, okay? All right, so if I'm looking for O negative?
Nothing.
Nothing, okay? So I don't have any antigens whatsoever. Okay, we'll do one more. We'll do a tricky one since you guys are so, must be because it's later in the day and you're finally awake.
Red Bull.
You're not really awake, you're just wired. All right. So an individual has B antibodies and D antibodies.
A negative.
What would their typing tray look like? Do we look for antibodies when we're doing blood typing? No. So what do you have to figure out from the antibodies?
The blood type.
What their blood type is. So since they have B antibodies, what's their antigen? And just go a little more slowly, Diane. Okay, A. And since they have D antibodies,
Negative.
they don't have the D antigen. Okay? So now we can take that information and they're going to agglutinate. The A antibody A is going to find the A antigens on their blood type. Okay? Will not find any B antigens because their blood type is A, and will not find any D antigens because their blood type is A negative. If you understand that last one, you're doing great. You got this process figured out. Okay? If not, we need to move on. See me during lab. Okay? We can go over it some more. The key thing to this is that even though we talk about antibodies, we are not concerned about the antibodies in the person's blood that we are typing. We are using these antibodies like magnets to search for the antigen on the person's cell. So when we find the antigen, we use that to identify their blood type. So that's why knowing that he had B antibodies, that has nothing to do with these B antibodies. All right? We're looking for the B antigen, and the fact that he has B antibodies means he doesn't have the B antigen. You would never have antibodies for an antigen that your cells demonstrate. Okay? So again, if that doesn't make sense during lab time, we can look at it some more. Okay, if you weren't here on Tuesday, the lecture notes came in early during class time, so they're over there somewhere on the counter. You can pick them up. So the lymphatic system. Obviously, it's named after the lymphocytes, but it involves more than just the lymphocytes. So it's a twofold mechanism. The lymphocytes are involved in the defense mechanism, all right? And they use what we call specific defense. As I showed here with the plasma cells, they are using their cells, their B cell or their T cell receptors to specifically recognize markers on other cells. And if those markers interact, then the B cell or the T cell is activated and starts making their molecules to destroy or clump together whatever was possessing the marker. So that's the direct role of the lymphocyte. The entire lymphatic system, however, consists of a series of lymphatic vessels and additional lymphatic organs. And the other function of the lymphatic system is vascular. You thought we were done with blood vessels. We are done with blood vessels, but we're going to talk a little bit about lymphatic vessels. All right? The purpose of the lymphatic vascular system, not the cardiovascular, but the lymphatic vascular system, is to return excess interstitial fluid to the cardiovascular system from which it leaked. And how did it leak? Remember the diagram I drew back with blood pressure in our vascular system? We have the arterial end of our capillary bed. And then we have our capillary bed, which is simple squamous epithelium. And then we have the venous end. And as you saw from lecture, when we first started talking about blood, blood is formed of fluid in cells. Okay? And in the fluid, we have proteins. And one of the major proteins present is albumin. The role of albumin is to maintain a colloidal or osmotic pressure within the blood. So we use the term hydrostatic pressure, which is the force of the blood volume and the heart's contractile force pushing against the walls of the blood vessel. Since we're on the heart side of the capillary bed, hydrostatic pressure is higher than it is on the opposite side because two things have happened. We're farther away from the heart, plus we're leaking some of this fluid of the plasma out into the tissues. So our blood volume is less, therefore our hydrostatic pressure is less. Does that make sense? Okay, the farther away we get from the heart, the lower hydrostatic pressure is. Remember we have this diagram? And that's why blood flows towards the heart. It flows down its pressure gradient. Okay? So we're farther away from the heart, plus we've lost some of that fluid pressure, so the volume force is going to be decreased. Then within the blood itself, we have these colloidal proteins, the albumin. Bless you. Now, at the arterial end, osmotic pressure is lower than hydrostatic pressure. By the time we get to the venous end of our capillary bed, because we've lost some of that fluid and the proteins were too large to leave, the protein concentration has gone up. We haven't added more proteins, all right, but we've lost fluid. So the relative relationship between the two has changed. It's like having water evaporate. You've got a bucket of salt water, you leave it outside, the water evaporates, but the salt stays behind and it gets saltier. Okay? So some of this fluid has been forced out of the capillary bed by that higher hydrostatic pressure. And so now our osmotic pressure, relatively, is higher than our hydrostatic pressure. So as a result, we pull some of that fluid back in. Osmotic pressure is a pulling pressure, okay? But not enough to completely equilibrate the value of what was lost. So some fluid still remains in the interstitial spaces. Now, before we talk about that fluid that remains, what would increase fluid loss? What would increase the amount of fluid that seeps across our capillary bed?
Increased pressure.
Which one?
Hydrostatic.
Increasing hydrostatic pressure. So too high an arterial pressure could do it. Too high a venous pressure. So if the right ventricle is weaker and we have right-sided congestive heart failure, it backs up into the right atrium and into the vena cavas and then into the veins. It's going to back up into our capillary system as well. Okay? And it's going to increase the loss of fluid across the capillary bed. Somebody standing on their feet all day, and they increase gravitational force causing blood to pool in the deep veins can increase that. A woman who is pregnant, all right, and the weight of the size of the uterus and the fetus pushing on the inferior vena cava is going to impede the return of blood to the heart, and so it's going to back up and increase. You talk about someone, I've got to sit down and put my feet up. It's not just to get the weight off, but that it helps to return the blood back to the heart. All right, so what about the osmotic pressure? What can change? How can that change and increase the loss of fluid? Decreasing or increasing osmotic pressure would increase fluid loss to the tissues. Maybe it is too early for me. All right, so if we have too much osmotic pressure and we have a higher pulling force, are we going to lose more fluid or less fluid to the tissues? Less. So if we have decreased albumin, then we're going to have less pulling pressure. We're going to lose more because we don't have the opposing force to the blood pressure. So how do we get decreased albumin? Where does albumin come from? Yeah, but how does it get in the plasma?
Liver?
The liver. It's a protein. So either the source of proteins to the liver, poor protein in the diet, or liver failure, liver disease, where it can't utilize the protein to make albumin. It's going to decrease the amount of albumin, and we have lower osmotic pressure. So we talked about kids in, starving countries or inner cities that don't have sufficient nutrition. They have skinny arms and legs and a big swollen belly. That's because of increased fluid loss across the capillaries of the peritoneal lining of the abdomen.
Is it only the kids or adults get
It's
that?
seen mostly in that, but yeah, you can see it in adults too. They just usually show the pictures of the kids when they're trying to raise money for charity. Cancer. Okay, so someone who has liver disease or cancer, ascites, is that large fluid-filled area. If they have increased pressure in their lungs, they can get increased thoracic fluid there. All right, so obviously, not all of us are running around with systemic edema, so there's a mechanism to overcome that.
So if you're dehydrated, would your albumin levels increase?
All right, the overall total weight of the protein would, but the relative amount in a given volume of blood would. All right, so we have extra fluid out here in our space. The rest of it's been taken up and carried back to the heart. So now we would have edema if we didn't have some other mechanism of picking up that extra fluid. Hence, we get the lymphatic system. So if we look at the tissue area, you guys don't have to worry about the embryology part, but let's look at this image right here. So here's the vascular system. Here's the arterial end of our capillary bed. Here's the venous end. And here we have the capillaries where we're losing and adding fluid to our extracellular fluid. The capillaries that are green are blind-ending lymphatic capillaries. They're still made of a simple squamous epithelium, but they're larger in diameter typically than the blood capillaries, and they're one way. Okay. Blood capillaries are open at both ends. Lymphatic capillaries are blind-ending. They have little filaments that anchor the edges of the simple squamous cell out into the connective tissue. And as increased fluid builds up around the blind-ending lymphatic capillary, it actually pushes apart these gaps. And that excess fluid enters into the lymphatic capillary. Right? So we find the two types of capillaries in the same area, but the lymphatic capillary is going to have just tissue fluid, and the blood capillary is going to have red blood cells and white blood cells. Now, the lymphatic capillary can have white blood cells too. Because remember, we have white blood cells, their site of function is in the tissue fluid. So as this gap opens up, fluid can enter in, but so can white blood cells. Histology, if we look at a cross-section or longitudinal section of a vessel, it might be a very thin-walled vein or a lymphatic. It might be a postcapillary venule or a lymphatic capillary. Only if it has red blood cells in it can you say it's a blood vessel and not a lymphatic vessel. Okay? Because if it has fluid only or fluid and white blood cells, you're not going to be able to tell which one it is. So the lymphatic capillaries join together and form larger vessels called lymphatics. And those join together and form still larger vessels called lymphatic trunks. And all of those flow towards the heart. Now, if we look at what it's picking up, if this happens to be extracellular fluid just deep to the lining of the stomach or the bronchioles, not only might it pick up white blood cells and fluid, but it could pick up a virus or some bacteria that have happened to cross between the cells of your digestive tract. And that's going to get carried all the way back to the heart. Not acceptable. Because blood is considered to be sterile, unless you actually have a blood infection. Okay? So we don't want to take all this tissue fluid and dump it back into the heart. There might be a metastasized cancer cell in there. We don't want to just put all that stuff back into the blood. So along the way, the lymphatic fluid passes through a series of lymph nodes. And lymph nodes are populations of B cells and T cells that create a filtering mechanism where the lymphatic fluid has to flow slowly by these lymphocytes and macrophages. And there's a very high opportunity of the bacteria or the virus coming in contact with a B cell or a T cell that recognizes one of the protein's structures on its surface. So this happens over and over and over as the blood flows back to the heart. Now you don't have to know any specific lymphatic vessels or trunks, okay? They're usually identified anatomically, so we have jugular, mediastinal, and we have iliac and stuff. I do want you, there's a few groups or locations of lymph nodes that I want you to know, like the inguinal lymph nodes or the axillary lymph nodes or the cervical lymph nodes, but those were terminology for body regions that you became acquainted with in 430, so most of you know where those areas are. There's an asymmetrical return from right to left side of the body. So all the lymphatics in the lower extremities and the abdomen join together to form a dilated vessel. Well, they form a vessel, the distal region of which is dilated, or I'm sorry, the proximal region of which is dilated. And that is known as the cisterna chyli. So lymphatics from the abdominal region that have joined in with lymphatics from the lower extremities and your organs all come in to the cisterna chyli. And then that narrows and flows. We find this channel right here, which is known as the thoracic duct. And it flows up almost the center along the surface of the bodies, the anterior surface of the bodies of the thoracic vertebra. And it is going to enter into the subclavian vein just about the point where it receives the internal jugular. So it crosses up and I've drawn this a little fat here. And enters into the subclavian on the left side, about the point where the internal jugular and the subclavian join together, all right? So this is going to carry lymphatic fluid from the right side of the head, neck, sorry, not right side, left side, of the head, neck, upper extremity, left upper extremity, left chest, and then all of the abdomen and both lower extremities. Sure would be a lot easier just to write leg, but that would be incorrect. Okay? So all of that is carried by the thoracic duct. You can see here, the blue are the zygous and azygous, so we're looking at the green. Here's the cisterna chyli, so running up here and going in right here. Okay. On the right side, we have a very short, here's the thoracic duct, we have a very short right lymphatic duct. So that's going to be entering into right here. It's about 3/4 of an inch to an inch long.
Thank you.
And we have, that will be the right lymphatic duct. Thank you, Diane. And it's going to be right head, right neck, right upper extremity, and right chest. Okay. So we have 2 of these lymphatic models. All of these fine light cream colored lines are showing the lymphatics. The yellow dots are the lymph nodes. Here is about a 1/2 inch long right lymphatic duct. And coming up here behind the portal vein is the cisterna chyli, and then you can see the thoracic duct coming up here and emptying into the left subclavian right here. I always have to smile when I get to this point of the lecture concept. When I was teaching at the medical school, one of the faculty that did this component of the system would always talk about, in the thoracic cavity there are 5 birds. There's the azygos, the hemiazygos, the accessory hemiazygos, the thoracic duct, and the right thoracic duct, or the right lymphatic duct. And so on their exams, the students had to choose between 2 essays in addition to their multiple choice questions. And I was grading one of them, and there's the student, it was the 2nd time through his 1st year of med school. During med school, they do everything they can to keep you there. Once you're in there, if you're in dental school, they kick you out as soon as it looks like you're about to fail a class. So it was the 2nd time through, and they were supposed to write complete sentences for their essay, and he started out, in the thoracic cavity there are 5 ducts. But no, there are no ducts in the human body. But that was the way he remembered it. You just couldn't transfer it to the actual anatomical structures. All right, so those are the only models we have that really show those components are this one, and there's a similar identical one in the back. Or I might show the lymphatic ducts or to you on a diagram as far as a lab quiz or a lab exam is concerned. So that's the vascular aspect of the lymphatic system, the vessel system. It has both components to it because it's returning the fluid to the cardiovascular system. And then we also have the defense component to it because of the lymph nodes filtering out any pathogens or cancer cells that might be present. Okay? So the next unit, we'll take a break in about 15 minutes since we got started a little bit late. The next section is looking at the organs now that function as a component of the lymphatic system. These are the major cells, obviously the lymphocytes, that are part of this organs. And of course, their purpose is entirely to do defense mechanism. And it is specific to defense. It's recognition of problem cells or viruses based on marker molecules on their surface. So the T lymphocytes and the B lymphocytes have specific receptors which recognize foreign cell markers, the antigens. Okay. Now, the process by which they recognize these is a little bit different. And we'll talk about T cells on Tuesday next week. We talked about B cells a little bit already because we talked about how D antibodies are produced. All right. But in either case, this is activated through the presentation of antigens. And macrophages, which are not specific immunity, they're going to phagocytose these foreign cells anyway, are part of the APC group. one of those primary antigen-presenting cells. That process we'll talk about on Tuesday. I don't keep promising this to you, all right? But that's why they're listed here. Because as the lymphatic fluid is filtering through the lymph nodes or the tonsils, not only do we have the B and the T cells, but we have high numbers of macrophages, not just to phagocytose those foreign cells, but to break them down with their lysosomal enzymes and to present them to the T cells and the B cells and say, this is the one. Well, they don't say anything. But in the random opportunity, the chance that might match their receptor and activate that T cell or B cell. Okay, so they're part of that process of activation. Not they will destroy, but that's indiscriminate. It's not, they don't have to recognize them to destroy them. All right, anybody remember what reticular cells are? Somebody this morning thought I was talking about the reticulocyte, the red blood cell that still has RER, and it's not that. A reticular cell is a cell that you all know very well by a different name. It's a special form of it. Fibroblasts. What three types of fibers do fibroblasts form? Collagen, elastic, and reticular fibers, which are really just, to make things confusing, a special kind of collagen. All right? So reticular cells are very elongated fibroblasts, and they wrap themselves around the actual collagen fiber. So we find these long structures, and I've got a picture a little bit, a really good picture, even better than that one. Here. So this is in a lymph node, and this is actually a sinus. It's not a high congregation of cells. It's a channel through which lymphatic fluid is flowing. So here's the lymphocytes, here's a macrophage, and see these long fibers? This is actually a fibroblast cell wrapped around collagen fibers. So we call the combination of the two the reticular fiber. It's really both. Okay? So the reticular cells function to entrap, well, it's not like they put a bait out, to hold lymphocytes together, otherwise they would kind of diffuse and percolate away. So they hold them in high concentration, and they also slow, because they crisscross these channels, they provide a bedwork for the cells to be located, and they slow the flow of fluid, making it more likely that if a T cell or a B cell does come in contact with an antigen that fits its receptor, that it has time to interact with it. And it's not just, oh, hello over there, you know, and I don't have time to shake your hand and hold on to you and make antibodies against you. So it slows that fluid down and helps to support and forms like a sponge-like network that the lymphocytes are found in. So all of these dots that you see here are lymphocytes, and holding them there are these reticular cells. And even where we don't have as many lymphocytes, these light areas right here, this is what this would be looking like. Okay? So there's still fibers crisscrossing that space. Not as many, but there are still some fibers there. So those are the major cell types that we see in lymphatic tissue. The T lymphocyte, and again, we're going to talk about their specific functions next week. There are 4 or 5 different types of T lymphocytes. It's the larger cell over there on the right. And then we have the B lymphocyte.